Hey everybody, welcome back. Today we're going to be discussing the cell cycle and we're going to start a little bit on mitosis. So the cell cycle is shown here. A cell is going to begin the cell cycle right over here, and then it's going to end the cell cycle after it gets all the way around. And the cell cycle is really just a description of what the cell is doing during this life cycle from creation to making daughter cells. And remember, a cell cannot be just created out of nothing. A cell has to be created from another living cell. So when it is created, it is here in this G1 phase. We used to call it gap phase because we didn't think a lot was going on, but really you can think of it as the growth phase because the cell is a little bit small because it's just been divided from a bigger cell. So it has to grow, it has to duplicate some of its organelles, make sure it makes more mitochondria, more endoplasmic reticulum so that it can do its day job. Now, some cells don't ever divide. Muscle cells don't divide, neurons don't divide, and so they're said to be in G1. And so really what they're doing here is they're just going to do whatever their job is, and they're just going to hang out. They're never going to go through the rest of the cell cycle. So we call this G0 because they're kind of stuck in G1. They're not going to proceed to another part of interphase. Our next phase, if we do decide that we're going to divide, is called the S phase. You can think of S for synthesis. And this is where we go through DNA replication. Now the cell has already decided that it needs to divide. And so in order to make two cells from one cell, we have to duplicate our DNA. That happens here in S phase. And then there's this other G phase over here, G2. And what's going on over here is more growth and more organelle replication. And there are some very important checkpoints that the cell has to go through. Before we allow the cell to replicate its DNA, we have a checkpoint right here, the G1 checkpoint. And we want to make sure, is everything okay with the cell? Do we have enough nutrients to allow the cell to divide? There's another really important checkpoint right over here, the G2 checkpoint. And that's the one where we basically look and say, okay, did DNA replication happen properly, or did we just create a Frankenstein cell? If everything's okay, the cell will be allowed to proceed into G2. Now, all of these phases, G1, S, and G2, are all part of interphase. So if you look at how much of this circle is taken up by G1, S, and G2, you can see it's quite a bit. It's actually more than is shown here. Most cells spend about 90% of their lives in interphase. They only spend about 10% of their lives down here dividing. This phase right here is called mitosis. So a lot of people think mitosis is a type of cell division, and we kind of use that word to mean it, but mitosis is technically the division of genetic material, basically DNA. So up here, the cell grew, we duplicated our DNA, we had some more cells growing and more organelles replicating, and in this part over here, mitosis, we're going to separate that DNA that we replicated. And then over here in cytokinesis, you see what I'm writing? In cytokinesis, that's the division of the cytoplasm and all the stuff that's in there. So the organelles get divided, all the goo that's inside the cells get divided. And so what we're doing here is we're going from one big cell with twice as much DNA as we need, and we're going to divide into two cells. And in mitosis, the two cells are genetically identical. So we went from one big cell and we divided into two little cells. And that's why, as those two little cells enter G1, they have to grow because they're kind of smaller. And it goes around again. And then each one of those cells could potentially go through the entire cell cycle and make 2 more cells. Now there's another checkpoint here, and that happens right about here in metaphase. And we are checking to make sure that all of the DNA is lined up properly and that both cells are going to get identical copies of DNA. So we have 3 different checkpoints within the cell cycle. We have 2 main phases, interphase, and then this one is mitosis and cytokinesis. Now, within mitosis, you'll notice your textbook actually gives you 5 little subphases for mitosis. I'm going to smush these first 2 together, and we're really just going to use prophase. That's how most other textbooks have it. So prophase, metaphase, anaphase, and telophase. You can think of them as PMAT. Some people like to say probably means another test. That helps you keep it straight in your head. Those are the phases of mitosis, and we're going to talk about those in the mitosis video. For right now, though, I want to talk about DNA replication. We're going to be talking about exactly how DNA replicates in another chapter, but for today, I just wanted to give you a little bit of update. So DNA is separated into structures called chromosomes, and a chromosome looks something like this. It has a little structure in the middle called a centromere. And all along the arms of the DNA, we have things called genes. And a gene is a section of DNA that specifies how to make a protein. And you have thousands of genes on each chromosome. Now, humans have 23 different chromosomes. So 23 different pieces of DNA in our cells. In our normal body cells, which we call somatic cells, we actually have 2 copies of each chromosome. All right, how do we get that? Well, 1 copy, say this is chromosome 1, came from your maternal parent. The other copy, here's another copy with another centromere and other genes, came from your paternal parent. So you have 2 different copies of chromosome 1. Now they're not identical. They have genes that correlate, but a gene can have more than one type of protein it specifies. So let's talk about the genes for eye color. You could have a gene that says blue eye color, or you could have a gene that specifies brown eye color. They both are specifying eye color, so they're correlated, but there's different versions of the gene, and those are called alleles. So the 2 copies of chromosome 1 that you inherit, one from maternal parent, one from paternal parent, have different alleles of the same genes on them. So your eye color gene might be here, and it would be in a very similar place on the paternal copy. You might have a hair color gene, and you would have a corresponding hair color gene on this one. This one might be blonde hair, this one might be brown hair. So what we can say is, even though we have 23 different chromosomes in our somatic cells, we actually have 46 chromosomes total. Now we call those cells that have 2 copies diploid. Di for 2 and ploid. All right, so diploid cells have 46 chromosomes. We also call them 2N because N is the number of chromosomes, and we have 2N, 2 copies of each chromosome. The number of chromosomes doesn't necessarily correlate with how complicated an organism is. For example, a potato has 48 chromosomes, and a hermit crab has 254 chromosomes. So this is just sort of a random way of splitting up all the DNA into manageable pieces. Now the chromosomes are numbered. We start with number 1, which is the biggest chromosome. Chromosome number 2 might be slightly smaller. The centromere isn't always in the middle. It could be on one side. So chromosome number 3 is going to be even smaller. I'm trying not to draw it in the middle. I'm so used to drawing it in the middle. But as you can see, I could draw out 23 of these, and then I would also have to draw out 23 of these. And so again, chromosome number 1 from maternal and chromosome number 1 from paternal are going to be what we call homologous chromosomes, which means they're not genetically identical, but they code for the same types of things in the same places, although they might have different alleles. Now remember, we talked about duplicating our DNA. This happens here in interphase. And the weird thing is we're going to come in with our 46 chromosomes, right? 2 copies of each chromosome, 1 from mom, 1 from dad. And we're actually going to leave S phase with 46 chromosomes. So how does that work? How do you duplicate your DNA and end up with the same number of chromosomes? Well, it's kind of a trick. So basically, when we duplicate our chromosome, we start out with our initial chromosome, number 1, with all of our genes on it, and our centromere in the middle. And we're going to make a duplicate copy, but we're going to make the copy stick to the first one. And they're stuck together still at the centromere. And when we count how many chromosomes we have, we actually count how many centromeres we have. So even though we have doubled the amount of DNA, we haven't doubled the number of centromeres because the DNA is still stuck together. Remember, we're not going to separate this out for quite some time. We're duplicating here, and we're not going to really separate out the DNA until we get to anaphase of mitosis. So we're going to do that to chromosome number 1 from your maternal parent, and we're going to do the same thing to chromosome number 1 from the paternal parent. And so our homologous chromosomes now, instead of being single, they're doubled. So here's another vocab word for you. This is called a chromatid. That's a single chromosome here. So this is a chromatid, this is a chromatid, this is a chromatid. These two together are called sister chromatids. They are genetically identical. These two are called sister chromatids. Also genetically identical, because we've copied one to make the other. We still know that chromosome number 1 from maternal parent and chromosome number 1 from paternal parent are homologous chromosomes. So these two are homologous chromosomes, not genetically identical. These two are sister chromatids, genetically identical. So if we look at what the chromosomes are going to look like throughout the cell cycle, before S phase, they're single. They're just single chromatids. But after S phase, they're going to be in this sort of doubled butterfly shape, and they're going to have sister chromatids. They're going to stay as sister chromatids until here in anaphase. And then we're going to rip the chromatids apart so that each daughter cell will get one sister chromatid from this pair. Okay, that's it for the cell cycle. We're going to move on to mitosis next, so come back for that.